So, I'm Yuexi from Xi'an Jiaotong University. Today, I will present our work HI-Synergy, a hypergraph and interaction-aware multi-granularity network for predicting synergistic drug combinations. This is a collaboration with Jianzu, Yongheng Sun, and Lou Xinzhang. Our model uses hypergraph structures and multi-level biological interactions to improve synergy prediction accuracy and interpretability. Here is a quick overview. I'll cover background, methods, results, and conclusions. Let's start with the background. So, what is synergy? When two drugs are combined, their effect can differ from just adding them. If the effect is stronger, it's synergy. Otherwise, it's antagonism. So, in this figure, the line here is the active line. The point below the active line means the combination achieves the same effect with less dose of each drug. So that is synergy, which makes treatment more effective and can reduce side effects. For the points here, they are antagonism. So combination therapies are critical in cancer treatment to enhance drug efficacy, reduce toxic side effects, and mitigate drug resistance. To recommend effective combinations, we need accurate synergy prediction models. And also another challenge is explaining the biological reasons behind these predictions. Our task here is to predict if a given drug pair and cell line will result in synergy. So how does our model predict drug synergy? The first question is how to accurately predict drug synergy. The model adopts a multi-granularity strategy. The coarse granularity module extracts global interactions among drug-drug cell-line triplets, and the fine granularity module extracts local interactions between drug pairs and between the drug and the cell line. The second question is how to improve interpretability of predictions. Our model adaptively identifies biologically meaningful substructures at different scales. Here is an overview of our model. It contains the first is the initialization module, and the second, coarse granularity module with hypergraph. Next is the fine granularity module with interaction-aware attention, and the last is the prediction module. Let's start with the part A, the initialization module. In this module, each drug is modeled as a molecular graph, where the atoms are nodes and the bonds are edges here. The node features shown in the table capture the chemical properties of each atom. And we use the graphismorphism network to extract the node features. The GIN is known for its strong expressive power, updating each atom features by aggregating information from its neighbors over multiple layers. We stick 3 GIN layers to the node-level representation, and we got the substructure representation. So after GIN, we will apply the SAG pool, which uses one-layer GCN to compute node importance scores. This helps aggregate key substructures at multiple scales and improve the interpretability of the model. And next is the JKNet. JKNet here fuses features from multiple GIN layers by concatenation. This allows adaptive combination of multi-scale information. And then again, we apply SAG pool here to get the final molecular level drug representation. So in the initialization module, we can get the substructure level representation of the drug and also the molecular level representation. For the cell line expression data, we use the MLP to get the cell line representation. And next, I will introduce the coarse granularity module with hypergraph. The input of this module is the two drugs molecular level representation and the cell line representation. In this module, we built a hypergraph where nodes represent drugs or cell lines, and hyper-edges connect drug-drug cell line triplets that exhibit synergy. We apply hypergraph convolution layers to capture high-order interactions among these nodes. So the hypergraph convolution updates node features, passes them to edges for aggregation, and then propagates the updated information back to the nodes. Residual connections from earlier layers help enhance the representation. The output of this module is a coarse-level global triplet representation. Next is the fine granularity module. The input of this module is the substructure level representation of drugs and the cell line representation of the cell line. In this module, we focus on local interactions at the substructure level. We use cross-attention here to model interactions between the two drugs and between each drug and the cell line. This results in 6 interaction vectors, which are fused using self-attention module here to form a unified fine-level representation, capturing the local drug-drug and drug-cell line interactions. Finally, I will introduce the prediction module. The input is from the part B and part C. So we combine the coarse level and the fine level representations and pass them through a highway network. The highway network uses transform gates here for nonlinear feature learning and the carry gates here to retain important row input, balancing new and original information. Finally, a two-layer MLP classifier predicts whether the drug pair is synergistic. So this is our model. To evaluate each module's contribution, we tested variants that remove the coarse granularity module, the residual connections in the hypergraph layers, the fine granularity module, and the highway network. This helps us evaluate how each component affects overall performance. Here is the result. This chart shows how performance drops when each module is removed. The downward bars indicate the decrease in performance compared to the full model. The blue line here is the full model, and we can see that removing the highway network causes a biggest decline across all metrics. Coarse granularity module here has a larger impact than fine granularity, where residual connections have minimal effect. And this slide shows how features representations evolve across modules. Initially, in Figure A, synergetic and antagonistic samples are mixed with no clear separation. And adding the coarse granularity module in Figure B introduces some structure, but separation remains unclear. In Figure C, fine-grained drug-drug and drug-cell line interactions improve the separation, and finally, Figure D shows the full model achieving clear class separation with well-clustered embeddings and strong classification. And next is our loss function. Our total loss combines 3 parts. The first is a classification loss, which we using the cross-entropy, and then the reconstruction loss, allowing learned hypergraph similarities with original drug fingerprints and the gene's expression data. And next is the contrastive loss, improving separation between positive and negative samples in the embedding space. Together, this helps the model learn accurate and biologically meaningful representations. And we can see that removing both the reconstruction loss and the contrastive loss leads to the largest drop in performance. This confirms that both components are essential for learning informative and discriminative representations. And here are our results. We use data from DrugCom and GDAC2 datasets. Inputs are drug molecular graphs and cell line gene expression data. The task is a binary classification of synergy versus non-synergy. And this slide presents 3 validation strategies. The first is the random cross-validation, splits all triplets randomly into 5 non-overlapping folds. And next is the leave drug pair out, which ensures that drug pairs do not appear in the training data. And the last is the cell line out test, that the model on cell lines will not appear in the training data. And we compare our models with 4 machine learning models and 7 deep learning baseline models, covering a wide range of synergy prediction approaches. This figure shows the relative performance improvements of our method over the best baseline under random cross-validation. And each bar here represents the performance gains across different metrics for drug comp dataset shown in blue and GDSC2 in orange. Our method consistently outperforms our baselines on both datasets. And this table shows model performance in leave drug pair out and leave cell line out scenarios on 2 datasets. Our model demonstrates strong generalization in both settings. And this figure shows how SAG pool identifies important substructures in anti-cancer drugs across layers, with redder colors here indicating higher importance. The top row for the first drug shows highlighting anti-cancer rings associated with DNA damage. And the bottom rows here for the second drug emphasizing key anti-cancer motifs. The results demonstrated that our model can identify substructures that are biologically meaningful. And this table presents novel drug combinations predicted by our model. Here are 12 ranked predictions, and here 5 are supported by existing literature in the same cell lines or tissues, and 7 are validated in other tissues. These combinations cover key cell lines from drug comp and 3 cell lines in GDSC2 dataset. The results demonstrate strong generalization and confirm the biological relevance of our predictions. In conclusion, we developed a multi-granularity neural network for drug synergy prediction. Our model combines global, hypergraph, and local interaction-aware modules to capture multi-scale biological interactions, and our model outperforms existing models. Substructure visualizations confirm the model's clear biological interpretability. Our future work will focus on adapting the model into a useful tool for clinical studies. And lastly, we thank fellow lab members at Xi'an University and the National University of Singapore. Thank you for your attention, and please feel free to ask any questions.
Great, thank you. Any question? We're, I guess, 5 minutes over the time. I don't know if. 1 minute. Okay, so I can ask you a question. It seems to be a really interesting work in terms of synergetic drug combinations. Have you applied this algorithm to, for example, the flip side of this would be, drug contradictions. So one drug is not supposed to be taken with another drug. Would you think, this particular approach might work there as well?
Yes, I think just we need to make the classification from the synergistic to antagonistic, and then we can solve this problem.
Cool. Any other questions? If not, let's thank the speaker again. Thank you.